All right, so we've started recording. So our plan for today and for Wednesday is to get through talking about amino acids, proteins, and enzymes. This is an important chapter to understand like how the body functions, and a lot of usually what targeted medication is about targeting proteins and specifically enzymes. So we'll spend 2 days on this, and then after that, we're actually only going to have 2 more classes. We have this week, which is I think our last full week, and then we meet next Monday, and that's it. We'll have the final, I believe it's set for Saturday, I think at 5 p.m. I'll have to double check that. I don't have the syllabus right in front of me, but that's what we're looking for. We're down to our last week and change. Once things are done, so once we've finished off chapter 17 next Monday, I will release a study guide. It will be a cumulative exam, so it will be all the chapters we covered. I know it's a lot, 17 chapters. So you can imagine I'm looking at big picture things, right? The most important concepts of each chapter will be covered. That's kind of the way it's going. The administration has asked us to finalize what the layout of the exam will be by tomorrow, and so I'll send that out to you guys as well. It won't be the questions, but it'll be the layout for the exam. So any questions on that before we get started into lecture? All right. So if you think of a question later, just email me. I'll get back to you guys. All right. So why don't we get started? So we're on chapter 16 now. And I know I asked this question a long time ago. I think it was like one of like the first days of class. But Haley, I think it was you who answered when I asked. It was an open-ended question. It said, what are proteins to people? And most people immediately think of like meat or like protein powder or like, I don't know what else. This concept of like food, right? Food definitely is made of protein, at least a part of it, right? But we're going to go a little bit deeper on that. We're not necessarily just talking about like food protein. We're talking about proteins that do jobs in the body, which there's around 20,000 different proteins in a human. We're just going to talk in general what are proteins and how are they made, and what makes them up and what are their functions, right? So we're going to expand our definition beyond just eating meat. So we'll start by talking about functions, right? So even though we have 20,000, this is just at least genetically, we haven't identified all of them, but you're giving that there's around 20,000 proteins, different proteins in the human body. These are all made up from only 20 different building blocks. And so they're all polymers. So most people think a polymer is like a plastic, and that is true, but a polymer just means a repeating unit in basically a straight line. And so proteins are just made up of different combinations of these 20 amino acids. And they can be as short as like 50 amino acids. For a protein, you can have peptides that are as small as 2. Or you can have enormous proteins like Titan. I don't know how many amino acids it is, but it's absolutely gargantuan, the protein. So you run the gambit in terms of size, right? How these 20 different building blocks are arranged are going to give each protein a different characteristic and different functions depending on the order that they appear. And we'll talk about the theory of how that order affects their shape and how that order affects their reactivity or affect their function. They have different types of functions. So typically proteins is kind of like an umbrella term. Enzymes are proteins. The difference are that when you just say protein, we're usually talking about structural components, things that don't participate in chemical reactions. Those usually are referred to as proteins. And so this will make things up like cartilage, which is, like your ear, your tip of your nose and stuff like that. Sharks don't have bones, sharks have cartilage. Your muscles are all protein, your hair and your nails are all protein, right? And those are just basically their structural function. Enzymes are the ones that actually do chemistry. So, you have enzymes that digest your food, enzymes that regulate your metabolism, enzymes that modify other enzymes so they do different jobs, right? So actually, the interaction between all the proteins in a cell is so complex, we haven't even come close to mapping the interactome. We've tried, we've got okay models, but we're nowhere near being able to do that yet. And then you also have other, very specific types of proteins. You've probably heard of hemoglobin or myoglobin, right? Their function is to transport oxygen in the blood to the surrounding cells. So let's move on from that. So this is just kind of like a summary of, the types of things that proteins do. We're not going to, I don't expect you to memorize each type. This is more just like to get a more holistic view, right? But if you go through and take a look at a bunch of names, they all have mostly similar endings. We're going to find out that proteins usually end in IN, so like keratin, but this is not always the case because obviously collagen, right, doesn't end in IN. But if we go through myosin, actin, hemoglobin, casein, ferritin, right, these are more passive proteins. They sit there and do things, but they're not necessarily doing, chemistry, right? Things that do chemistry usually are ended with ACE. So if you see a protein and ends in IN, it's probably structural or transport or storage. If it ends in ACE, it is almost definitely always an enzyme, and that means it's going to do some sort of chemical reaction. And so we're going to focus on both. We won't touch enzymes at the very end because they're very specific, but we'll start talking about what are proteins. Before we jump into like really getting in what a protein is, if you don't have a picture in your head, it's going to be kind of hard to build up. So what I want to show you, and I'm going to switch screens, is what chemists talk about when they're talking about like protein structures, right? And I noticed that it's not just meat, right? We're talking about teeny tiny molecules. So what I have here, and I'll do it just a little bit on this, is the crystal structure. So that means you've literally crystallized the protein and we shoot X-rays at it and we have ways to figure out the structure from there. It's math that I can't do, but my friends can. They did their PhDs in this. So when we talk about proteins, we're talking about small molecules. Well, macromolecules, technically. They're still teeny tiny though. I did the math out. If a protein, I just took a generic size protein, and then kind of blew it up in terms of like scale to something that we would interact with. So the way I would say, if a human cell is roughly the size of Boston, a protein is about the size of a car, a small car. And so to give you perspective, like how small a protein is in regard to how large a cell is, right? You cannot see a protein under the microscope, but you can see a cell under the microscope. So just to give you a sense of how big they are. So what you see here is the protein insulin, and it doesn't look anything like you probably, anything we've looked at so far, right? It looks like there's a bunch of squiggly lines, right? But this is how chemists and biochemists think of proteins. So what I have on right now is called the cartoon drawing. I know it sounds dumb, it's an old name, but we call these cartoons because it's just a representation of the structure. And we'll get into, maybe not today, but what we're going to name certain features. Like you notice there's helices in there, or you notice these big arrows, right? We'll talk about those a little bit later. That's going to be called secondary structure and tertiary structure. What I want to turn on, though, is let's change the representation of the polymer. So instead of cartoon, why don't we go to ball and stick? So I have them overlaid right now, and actually what you're seeing now is all the atoms and all the bonds inside a single protein. Looking at all of them, because you're talking about hundreds of atoms in one compound, it becomes very difficult to like visualize. That's why chemists and biochemists fall back on using the cartoon representation. So this is going to be part of your project at the end, right? Project app. Somewhere someone's got music on in the background, not muted. Hold on one second, guys. I can. Oh, all right. There we go. It's nice being the host. I can mute people. All right. If whoever I muted, if you need to talk and it's screwed up, just let me know. I'll fix that. All right. So, right, we're saying that we have hundreds of atoms in these representations. Oh, I know what I was saying. We're talking about the project that's coming up, right? Mini project. Like I said, part 1 you guys could have done already. Part 2 will be going to this website, and I'll do more of this on Wednesday. But you'll be using like this little feature just to play around. And I'm going to have you guys look at SARS-CoV-2 proteins. Like this is just human insulin. But again, play around with it, get a sense of what proteins are. Oops, I muted that. Right, so if we go back to our representation, turn off the ball and stick, I should be able to do that. All right. Oh, there we go. Remove representations. All right. Well, I'm done.
When is that project due again?
It's due May 6th, so it won't be due for over a week and a half.
Okay, thank you.
Yep. And if you haven't guys noticed, I don't know if I emailed you, I forget. It's already up on EC Learn. It was up on Friday. It's under one of the files. It just says mini project. All right, so that's what I want, just at least get a picture in your head of what a protein looks like before we start moving on and talking about how does the body build up and like make a protein, and then what do they do? So proteins are made out of amino acids. Oops, I don't know why. Oh, my iPad's super delayed today again. I don't know why. All right, so proteins, even though we saw how big that molecule was, right? We said it's made out of hundreds of atoms. We also said they're only made out of 20 individual unit blocks. And each block is called an amino acid, right? And so when we look at the structure, in general, what I have down here, this protein right here is alanine. We don't really need to worry about that right now. But this is the most generic. I mean, almost all amino acids look exactly like this. I'm going to highlight everything that does not change. All amino acids, all 20 of them, anything that I've highlighted in yellow doesn't change. The only thing that changes the amino acid's function is something that we call the R group. And we're going to notice that there's 20 different R groups that we talk about. In your body has ways of modifying them. And so even though we only have 20,000 proteins, there is so many modifications that usually you deal with that in grad school, and you become very selective on the type of modifications you study. So we're not going to get into that, but there are more ways your body can like make these amino acids a little bit different and change their functions. Another aspect, so that's the only thing that changes is that R group. Things that don't change, I highlighted them, but the more important bits are going to be, this is where the name amino acids come from. On one end, there will always be an amine, and we'll talk briefly why I have a charge on it when it becomes ammonium. It's just because in water, amines typically form ammonium, but you're always going to have an N terminus or an ammonium group, and that's what we see up here. And you will always have a carboxylate group. That carboxylate group is always at the end, and we call it the carboxy terminus or carboxylate group. That leaves the middle carbon. The reason it's called alpha is because it's the first carbon if you read left to right. It's the only reason. The second carbon, carboxylate one, it's called beta. We're not going to get into beta amino acids, but they do exist. But in general, that's the structure of an amino acid. So why don't we take a look at the structures of the 20 different amino acids, right? So they're going to roughly fall into 2 classifications, and then polar is going to expand into 2 other classifications. So it's more like an umbrella type feature. So the 2 big groups are going to be nonpolar, and these are going to be hydrophobic, which we know means water hating. This is why proteins actually fold up into balls, is because those proteins that hate water, well, if they hate water, they like other nonpolar molecules, and they'll actually wrap themselves into a ball and then surround themselves with polar amino acids. And that's how they actually get their structure and then also stay soluble in water. So usually nonpolar amino acids are more for structure of like the overall structure, though they can participate in other interactions. Polar molecules, right, these are going to be hydrophilic, which means water loving. These are usually on the outside, not always, but usually on the outside of proteins. Hydrophobic is usually on the inside. Again, that's just a generic, it's not always the case, just usually, right? So if we just take a look at 2 representative examples, the first being valine, which we see here. So like you see highlighted in blue, it's everything that I showed in that last slide that doesn't change. The R groups are in yellow. And so when we take a look at valine, valine only is made out of carbon and hydrogen. They're nonpolar. They're not going to be wanting to mix with that polar solvent of water. So valine is nonpolar. Asparagine, which you see to the right, we can see that we have this amide group on the top. And so I want to take just a 2-second pause. Well, actually, let me finish my thought, and then I'll explain amines and amides, because I know we had to kind of brush over that. in the chapter, I think that was 14, right? So I want to make sure we just remember what that is. But for now, it is an amide group. Amides are soluble in water. We know we have a nitrogen and an oxygen. Those are very electronegative. That makes it polar. That makes it want to interact with water. So we can see very distinctly, we have a nonpolar amino acid valine and a polar amino acid asparagine. So let me take let me pause on that before I go into amines and amides real quick. Are we good so far on what we've covered? Anything that I should clarify? All right, I'll take science as a good thing. So two things that I want to talk about real quick are going to be amines. And amines are anything that it's a carbon-containing compound. Again, I'll make that as a squiggly line. And it has an NH2, an NH, or just an N, usually, with nothing else bound to it, right? So this is going to be our amine group or amine group. When amines go into water, they usually act as bases, and I'm running out of space to draw this, right? But if the amine is a base, that means water is an acid, and it will donate a hydrogen to the amine. In our take-home message right now, right, is that it would form, sorry, it's a little bit messy, right? That's gonna form a ammonium, and then it leaves behind hydroxide. So I know it's a weird way that I just drew that. Oops. But I'm trying to use the space that I got. But what I'm looking at is I started with an NH2, I dissolved it into water, and then you end up with an NH3. And that's why you're seeing what usually, if you Google it, you'll see it as written as usually NH2. When an amino acid is added to water, that amine immediately converts to an ammonium, so NH3+. So that's why we're seeing that charge. On the other side, it's an abbreviation, but if we look at the explicit structure, it's actually a carboxylic acid, right? And we already know when carboxylic acids are added to water, right, that H is going to dissociate, leaving behind a carboxylate. And that's exactly why we see here. You add the carboxylic acid to water, becomes a carboxylate. Amino acids are just, they're a class of compounds called Zwitter ions. We don't worry about that, but yeah, there is a base and an acid on the same molecule. When it's added to water, both interact with water. One is a base, one is an acid. They're going to be charged. All right. So I know that was a little bit quick. I have the slides up for amines and amides if you want a refresher on that. But the last thing I'll say is just to make sure we recognize what an amide is. An amide looks a lot like a carboxylic acid. The only difference is instead of having an O, you have an N. And then we're going to make sure nitrogen has 3 bonds. So an amide typically, wow, my computer's really slow. There's 2 should be showing up in a second. Come on. All right, I don't know what just happened. It should be NH2. I can see it on my laptop, on my iPad, but I'm not seeing it on my computer screen. So sorry about that. But that is our amide group. So the difference it makes when we're looking at an amine versus an amide, and man, it's really delayed, right? So you're gonna have to go back into your notes, right? The NH2, when I was talking about amines, does not have that carboxylate carbon. That's the biggest difference. All right, if this iPad's going this slow, I might have to, okay, there we go. So now that we kind of have the general structure of what an individual amino acid looks like, and it is divided into these groups, right, nonpolar and polar, so let's start taking a look at the molecules we have. And so what I'll do is I have to put this together, but I'm just going to make a cheat sheet for you guys, like most biochem students do this anyways, of having a list of all 20 amino acids with their name and structure. So you'll have that, for the exam. I'm not going to force you to memorize all 20 amino acids. So you should get familiar with them because as you move forward as in your career, most people are going to start to expect you to just know what they're talking about. So, but if we take a look at the groups, we start with the simplest being glycine. The R group is actually just a hydrogen, so it usually doesn't participate in any reactions. So that's, we can get to very specific reactions, like radical reactions, but we're not going to get there. But we just have an H for an R group, and we start to build up in complexity. Alanine, valine, leucine, isoleucine, right? And we're seeing this pattern. It's either made out of just hydrogen or carbon and hydrogen, making it hate water or hydrophobic. We can also have more complex structures rather than just straight chains. We can see we have phenylalanine, methionine. Methionine does have a sulfur in the middle. We have proline, which is an interesting amino acid because it actually wraps around itself and rebinds with the backbone. So that usually makes things pretty rigid, and it's actually how the nature has evolved to kind of direct things. Basically, it makes it not move. So it's kind of like locking things into place. But we can again see it's all made of carbon and hydrogen. And then we have tryptophan. Tryptophan does have a benzene ring, and it does have that, that nitrogen, but there's so much carbon. Basically, we're saying that little bit of nitrogen is basically it's not compensating enough for having so many carbons around to make it nonpolar. If we take a look at the polar amino acids, it's a smaller group, but we can take a look what makes them polar, right? Serine, right off the bat, yeah, I have a carbon in there, but I also have a hydroxyl group. That's going to make it soluble in water. Threonine, we can see we have the hydroxyl group, soluble in water. Tyrosine looks a lot like phenylalanine, but it has that top hydroxyl, soluble in water. Cysteine has a sulfur. That's a little, we didn't get into it when we were talking about electronegativity. Sulfur's on that borderline, so it actually does make it soluble. Asparagine, we saw already, has that amide functional group, makes it soluble in water. And then glutamine, all it is one additional carbon with an amide on the top, soluble in water. And so you can see it's basically the little changes in the structure is actually going to have a big deal at how they interact with others in space. And like I said, when we get to secondary structure, we'll talk a lot more about that. The last two groups are also polar, but it's going to be taking those polar groups and then dividing them into acid and bases. So it's almost like if we're making a flow chart, right? If we have amino acids, it's either going to be non-polar, and that pretty much ends that one, or it's polar. And then that kind of breaks into, I'll call neutral, which is the last slide we did where nothing was charged. My God, it stops. Okay, there's just a huge delay. It's either going to be acidic or basic. So what we've looked at right now, 2 slides ago was nonpolar. The last slides were neutral. We'll talk about acid next, and then basic after that. So it's kind of like a flow chart. And we only have 2 acidic amino acids. The first is aspartic acid. Has a similar name to asparagine, because if you look, the only difference is asparagine has an amide instead of a carboxylate. And then glutamic acid or glutamate. Again, it looks a lot like glutamine, which used to have that amide. That is now just a carboxylic acid. So it's changing its name to glutamic acid or glutamate. So if you've ever eaten monosodium glutamate, that's exactly what you're eating. It's just an amino acid with 2 sodiums on it to balance out the charge. That's it. And it's not toxic. I don't know where it got that bad rap. All right, so the last group are going to be basic amino acids. So maybe what I didn't well, I can go back here, right? What we're noticing here with the acidic ones, all of the R groups are carboxylic acids. That's why they're behaving like acids. All the bases are amines. And that goes back to a couple of slides ago, right, when I mentioned that amines, when added to water, act as a base. So they take a hydrogen from water, and then they gain that extra H that gives them a positive charge. And so this is a good slide to take a look at and to talk about primary, secondary, and tertiary amines. Just like when we were dealing with primary, secondary, and tertiary alcohols, right? We're just asking how many carbons are attached to that nitrogen, right? Or how many bonds rather, right? So if I look on, say, histidine right here, right? We notice this nitrogen in the middle, and it's kind of small, so I try to do my best, but we got 1 bond there, 2 bonds there. We have a tertiary amine. We take a look at the top up here. There's only 1 carbon attached when we're looking at lysine. There's our primary amine. And then we take a look at arginine. Arginine has a very interesting functional group. It's really soluble and actually can make a tremendous amount of connections, and they're pretty important. I actually don't know the name of that group that has so many nitrogens attached to that carbon, but if we definitely can see it definitely is going to be soluble in water, and then it has that amide group that's going to, or that's the amine group is going to act like a base. And so we take a look at that nitrogen. We have 1-2 bonds. So we have our secondary amine. And that's all we're looking for talking about the degrees of amines. But again, we can see all of them are soluble in water and then all behave as bases. So we have our histidine, our lysine, and our arginine. And so that actually gets us to a total of 20 now. And so actually this is one thing, one among a lot of things that people that are used to back up the theory of evolution, is that every single organism that we've ever encountered uses almost all 20 amino acids. And every organism we've ever encountered uses RNA or DNA or both. So it's basically saying that it gives credence to the idea that there's one, you know, the furthest common ancestor, I forget the term, but basically everything behaves with the same code, with the same structures, with the same building blocks, all of life as we know it. So that's why we can talk about, you know, these 20 amino acids, and it pretty much covers almost all organisms you'll talk about. Some organisms can modify them, and they might use some other strange amino acids. Like I know in tea, it's not built into the structure of proteins, but if you ever heard of theanine, if you get fresh tea, theanine is an amino acid that you can drink. It's good for your health. It has psychoactive effects, which is usually a relaxation thing. So that's why like fresh tea usually gives you a relaxation effect. It's because of the theanine has a psychoactive effect. Very subtle, but it's there, right? So if we have these 20 amino acids, right, we're going to have to practice drawing them, right? And so we kind of use something similar to a Fisher projection like we did with the carbohydrates. So what we do is we're going to put our carboxylate terminus on the top, right? Oh, sorry, not the carboxylate terminus. Sorry, we're going to put the R group on the top, and that's what we have here, because aspartic acid has that, well, acid, and it's a carboxylate group. We put the alpha carbon directly in the center, and then what we do is we spread out the ammonium group, the N terminus, and then the carboxylate group, the C terminus, left and right, and then the last H points down. I alluded to this a long time ago. I mean, it probably didn't sink in because we weren't exactly talking about this, but amino acids also have L and D isomers. We'll talk about that in a second. But in general, if you notice both that I have here, we're just taking the structure and we're just flattening it out. And the convention is that the N terminus is the beginning and the C terminus is always the end of a protein. So if you ever hear me say N terminus, that means the beginning of a protein. C terminus means the end of a protein. Wait, did I say that right? N terminus is the beginning, C terminus is the end. And so we'll do a little bit of practice with that, right? So if we took something like, let's say alanine. I know you don't have the sheet right now. I just happen to know them off the top of my head, right? But I know alanine's our functional group. All it is a methyl group. So if I follow my generic structure of how to draw amino acids, well, I can put my alpha carbon in the center. Just like when we read left to right, I'll put the beginning, quote unquote. We haven't made a chain of these yet, so there really is no beginning and end. But you'll see in a second why I keep mentioning it, that it is the beginning, right? We're going to have our amine group. You can put the hydrogens on the left or the right, both are fine, right? We have 3 of them. It'll be positively charged because it's in water. We'll put our carboxylate group. You can either draw it out explicitly like this, that's fine. If you want, it's a little bit more condensed, is to write COO minus, right? Because COO minus. That's where we're getting that from. We have that leftover H to make sure carbon has 4 bonds, and then we'll attach our R group. For alanine, the R group is just a methyl group. So in general, honestly, the only things you really need to memorize about drawing amino acids is it goes N to C, and then the R groups get attached to that, that alpha carbon in the center. That's it. Everything, anytime I ask you to draw something else, you're literally just changing this point. How do we feel on that? Do we want to do a little bit more practice, or does that seem reasonable?
Could we maybe do one more example?
Yeah, sure, let's do that. Let's do valine. I'm just trying to do, or you know what, let's even do that. We can just do threonine. Let's do that. Or serine, that's the one I wanted to do. Serine, so serine is another amino acid, it's polar. If we look at the R group, what it's going to look like, we'll have a CH2 and then an OH. And remember, I've memorized these, I've used these for a decade, so I don't expect you guys to know them now. But like I said, I will give you a sheet that will have all of the R groups, and it will be, be a little bit easier to put things together, right? So again, I can set up my generic structure. Pretty much nothing changes from this. I'm now just gonna make sure everything's in the right spot. I know my N terminus will have an NH3 plus because it ionizes in water. I know my C terminus will be a carboxylate, so I write it as COO minus. That H is down there just to make sure I round off, I have 4 bonds to carbon, and then I attach my R group. My R group for serine is going to be a CH2 and an OH. I can double check, every carbon, everything below here we've already double checked to make sure we have enough bonds, but to make sure everything's written correctly, right? The carbon has 1, 2, and then it's implied to have 2 more bonds to H, so that's 4. If I drew that out explicitly, right, it would look something like that. We know that the hydroxyl group, I can just write it as an OH, but if you want to be super specific, that oxygen should always have 2 bonds, and so we're in good shape. And so that would how we would draw serine. All right, so what I meant this slide to be empty for is to show you one other way to draw amino acids, and I'll do both real quick. So that's the explicit structure. I want to make sure you also see the condensed skeletal structure as well. And so that's what I want to show you. So I'll do that with both with alanine and with serine. So I'm going to cough for a second, guys, so excuse me. So if we're going to draw an amino acid that has the skeletal structure rather than that flat projection, we start the same way. We'll have on the left-hand side my ammonium. My alpha carbon's just represented by a change in angle. And then I'll have my carboxylate. So I still have my alpha carbon there. It's just drawn in skeletal form. And then if it's alanine, I add in my methyl group. There we go. If I do the same thing with serine, and we're seeing this way is a little bit, at least for me, it's much clearer and easier to see what's going on rather than having a bunch of H's hanging out. But that's both what we just showed you, the explicit structure and then also this condensed skeletal structure. I don't think the book goes deep into the skeletal structure even at all. It's just, this is a common way that you'll see things. And I do plan on some of the reactions, and I'll show you in a couple slides. I'll probably use skeletal structures because it'll be easier for you to see what's going on, because you'll see on the slide, I'll have explicit structures. It does get messy pretty quick, but then I'll clear that and then we'll look at skeletal structures. So I want to do both to make sure people are feeling comfortable and following what's going along. All right, so I'll give you guys, actually, you know what? I realized if you don't have the sheet in front of you, it's going to be guess and check. So maybe I'll just walk through this study check with you guys. The way I would attack this, if I had a question, it says, identify each of the following amino acids as either polar or non-polar. So it's not even asking if it is acidic or basic, right? So the way I would check, forget everything but what's attached to the alpha carbon, right? So I see glycine's a little weird, right? We did say it's R, I'll get rid of those, right? We did say its R group is just a hydrogen. It's gonna hate water. So I'm gonna make the reasonable conclusion that this is nonpolar, because that R group has nothing that is electronegative, something that will want to interact with water. Threonine, on the other hand, that R group, even though, yeah, I got 2 carbons on it, that R group has that hydroxyl group, that alcohol group, that immediately is going to make it polar. And so let's check to see what the answers say. Nonpolar and polar. So I know that was a kind of an easy one, but also at the same time, you don't have a list in front of you right now. But did it make sense how I attacked that, or was I not clear?
Can you explain the non-polar one again?
Yeah, maybe glycine's not the best example, but it's the idea when you're looking at the R group, and that's why I said I have to kind of like scribble out this H here and make it explicit. The R group technically is a hydrogen, and I'll put the other regular H just in black. Or actually, I can do this. Keep it similar. So I'm just going to scribble out that 2, right? So I drew the R group as just an H. So anything, if it's only made out of carbon and hydrogen, is going to be nonpolar. If you're looking for polar, it's either going to have an oxygen, a nitrogen, and then sometimes sulfur. It depends, because methionine has sulfur, but it's nonpolar because it depends on where it is, and that's something you're just going to have to recognize. But then you have something like cysteine, where the functional group is just a thiol, and that is polar. So it is going to matter a little bit on the structure, and I'll tell you the 2 off right off the bat that get a little confusing are going to be tryptophan. The reason for that is because in the structure, there's a nitrogen there, but it's also very big and bulky and a lot of carbon. So I'm saying that might throw people off seeing the nitrogen and immediately thinking polar. So you got to recognize tryptophan doesn't fit that bill. And then the other one is going to be methionine. I ran out of space, right? Tryptophan, W, and C. We'll talk about abbreviations later, but actually, it's not doing that now. Tryptophan and methionine. Those might be the only 2 that throw you off. Everything else follows the general rule. Non-polar is just carbon and hydrogen. Polar contains oxygen, nitrogen, or sulfur, with the 2 exceptions of tryptophan and methionine. Does that make more sense?
Yes, thank you.
Okay, good. All right, so I'm going to leave this up to you guys because pretty much it's just copying it directly over from the slide, that the handout that I'll give you. I have to finish making it though, but I can show you real quick. This is going to be another thing you're just going to have to get used to. It will also be on the sheet, and the book also discusses this, but the 3-letter code and the 1-letter abbreviations, this is just something you're going to have to memorize. It will be on the sheet I give you. Usually they're not too bad. Some are really bizarre though. Like glutamine is so we'll put like alanine is A, cysteine is C, right? But then you get glutamine is Q, glutamate is E, right? So there'll be some weird ones out there. But let's just do, we already did, let's do cysteine. We don't have to do one of these. I think we've seen valine a couple times now. But if I was going to draw these out, again, left to right, I have my ammonium connected to my center alpha carbon. I have my carboxylate. That H is just hanging around. And then if I looked it up, the cysteine functional group is just a thiol. The 3-letter code for cysteine, and this is again just something you'll have to just get used to, it will be on the chart, is cis, and the 1-letter code is C. Just so a heads up, like I'm saying, most of these are easy. Valine, 3-letter code is Val. 1-letter code is V. So a lot of them are easy. Some are weird. If I was going to draw cysteine, say, in the condensed skeletal structure, let me do it in a different color so it stands out. Right, we still have that ammonium terminus, the N terminus. We have our alpha carbon. Then we have our carboxylate. And our, things are blowing out like crazy around me. And then we have our R group, which is a thiol. So again, this is, I think most are going to come with practice. And there we go. So I alluded to this earlier. I was getting a little head over my skis, but when we have these amino acids, right, we haven't looked at putting them together into a peptide or into a protein. But I mentioned this before when we were talking about L and D isomers with carbohydrates, is that your body can recognize the difference. And I think I specifically used the example that D amino acids will set off a immune response because humans don't use D amino acid, don't use D amino acids. Some bacteria do in their cell wall, and then that is used as kind of a signal to the body that there's a bacterial infection in some cases. So how do I tell if I have an L or a D isomer, right? What I need to do, just like when I was looking at determining if a sugar is L or D, you look at the last chiral carbon, which will always be the alpha carbon for amino acids, so at least that's easy. And the rule is exactly the same. My hydroxyl group points to the left, I have a D amino acid. If my hydroxyl group, I'm looking at the wrong thing, sorry. These are glyceraldehyde. These are the sugars still. What I really want to focus on was the amino acids, right? These are what we've done already. Glyceraldehyde, I think I even used glyceraldehyde on the exam, right? But if we look at the amino acid, again, we're following the same idea. There's our alpha carbon in the center, right? Where is the ammonium group pointing now is going to tell me if it's L or D. If it's on the L, it's left. If it's on the D, it's dextro. So we're following the exact same rules. For carbohydrates, you follow the hydroxyl group. For amino acids, you follow the amine group, or that NH3+. So hopefully nothing too different, literally the same exact rules. And so this will be the last slide, not for today, because I want to just start to talk about primary structure, because there's primary, secondary, tertiary, and quaternary. We'll get through those. But just to kind of go on the same vein when we talked about like essential fats in the last chapter, that humans have evolved to say, well, it's easier for me to eat another animal or another plant and take the fat from them than it is to me for always spending that energy to make it myself. And so we do the same thing with amino acids. The human body can only synthesize 11 of them from scratch. The other 9 amino acids absolutely must be eaten through proteins and diet. So usually lower organisms and plants still hold on to the machinery to make these amino acids. So that's why you still, again, you eat for energy, but you also have to eat for nutrients. So like there's certain fats that you absolutely cannot make yourself, so you have to eat them. There are 9 amino acids that you absolutely must eat, or you will actually start to get very sick, because what your body will do is start to break down other proteins to try and cannibalize any of the leftover amino acids it can't synthesize. And so it's funny, I don't know, you guys probably seen Jurassic Park, right? When they talked about, they put a genetic mutation in the dinosaurs, so they had to be fed lysine. Yeah, that could work. Humans can't make lysine, but at the same time, all you have to do is eat something that does make lysine. And so that was pretty dumb fail-safe mechanism. All right. So I know we just went over a lot in 2 sections. Again, I know everything seems like a lot because this is an accelerated course. So take-home messages of what we were looking at, right? What is an amino acid? Recognizing that the R group is important, and that basically defines its function in the body. We've learned how to draw amino acids, right? And we've learned to distinguish if they're L or D amino acids. So now what we're going to move into is talking about primary structure. And this is going to be talking about taking 2 amino acids and then reacting them together. And then, once we get beyond just reacting 2 together, we'll eventually get to, in chapter 17, about how your body puts together hundreds of amino acids in a row. And it does about 1 per second. It's pretty quick to make a protein in your body. So let's talk about hooking up these amino acids to start to make a peptide. So a peptide is a very particular type of bond because it only happens between amino acids. Everything else you'd call it an amide bond, and usually you hear scientists call it amide bonds anyways, but peptide is also another way to say the same bond. It just particularly is referring to an amino acid amide bond. So how does this happen, right? So what I have here, I've taken glycine. How do I know it's glycine? Well, the R group's an H. And alanine. How do I know it's alanine? The R group is a methyl group. Now we'll get to see why I kept kind of falling back into old patterns of saying like the N terminus or the C terminus, when it's like, well, you only got one of them, right? The N terminus, and this comes out of how humans synthesize proteins. This is just the beginning of the protein. And again, we're reading left to right. The C terminus of the first amino acid, which is my carboxylate, is going to react with the amine of the N terminus of the next amino acid. So what ends up happening, I got a lot going on, so let me wipe that, right? A bond is going to form between these two. And actually it's going to form water. Where does it get it? Well, it actually pulls everything from the carboxylate and from the amine group. So we actually can see it's in bits and pieces. And it happens, and it's not as easy as just clicking together. There's multiple steps that happen, but effectively it's gonna pull off those two H's and the O and make water. And then that's what you see over here. So if I pull those atoms off and and things do rearrange, what you'll form, you used to have a carboxylate, you used to have an amine. When everything's said and done, you now have an amide. So we have a carboxylate, and we're getting that from the C terminus of my glycine. We have an amine, technically in the form of ammonium because it's in water, because it's protonated, but it's still an amine. We're getting that from the alanine, and then when they form this new bond, we form a peptide. And that peptide can also be called an amide bond. Sometimes you hear people say amide. It's funny because I go back and forth between saying both. Depends on whatever words I'm using, right? So we end up with this new peptide. It's a dipeptide because there's two of them. How do we write down the name? Well, it's actually pretty easy. We started with glycine and alanine. The first one in order that they appear, so that's the ones coming from the N terminal end. Instead of having glycine, just like we've seen a lot of times with the alkyl groups, you just remove the ending and put "cyl" or YL. So instead of being glycine, it's glycyl. And then alanine, you don't even change that. So it's just glycosalanine is going to be the name of that peptide. If I draw it out with the three-letter code, because it's a little easier to write, glyala. And then if you're going to use the one-letter codes, it's just GA. So take-home message here on this slide. Knowing what sides are reacting, right? It's always the C terminal of the first amino acid reacting with the N terminal of the second amino acid. They form a peptide bond, and then would you be able to name that peptide bond? Glycosalanine. And we'll do another one when we have a tripeptide to see how this starts to all flow together. Let me see if I can find a good place to stop. I think, yeah, let's stop at drawing peptides. So we'll get through 2 more slides. We can do that in 2 minutes. So all I have here is just kind of showing a little bit. It's exactly the same peptide I showed in the last slide. It's exactly the same reaction. All it is trying to show you in 3D space what's happening. There's my C terminus from the glycine. There's my amine from the alanine. And what did they do? They formed the amide peptide bond in that glycosalanine dipeptide. So if I was going to draw that as a skeletal structure, right, and then we'll stop at this and we'll call it a day. I have my N terminus from my glycine. If you want, you can just put an H. Sometimes it helps. Usually I would draw glycine just like that with the carboxylate, but I know I'm forming the dipeptide, so I get rid of that N, that O. It gets replaced with an amide bond. Usually, though, this is where it gets a little bit weird, I think, for some people, is that the R group alternates up and down in the skeletal structure. So it's just the way to kind of keep this, how we usually did, if I draw a long carbon molecule, I do this saw shape. It's to keep that saw shape. And it helps. And actually, it's not just because it's easier to view. It's because in real life, they do alternate left and right. If you followed it, like if you put it in a straight line, the R groups all point in different directions. They all don't point on the same side. So what we can do is, I know we ran out of time. Well, we can pick up, I know I just did this one too quickly probably. We can pick up with this as the starter slide on Wednesday, but I think we're in good shape. So I would say enjoy the day, but I don't know if it's gonna be one of those enjoyable days around here. I think sun is coming on Wednesday. So, all right, if you don't have any questions for me, you guys are free to head out. If you do, I can stick around for a little bit, but again, I gotta prep for my next section.
Thank you.
Yeah, no problem. See ya.
Thank
All right, thank you.
Yep, you're welcome.